Hello, everybody, and welcome. We hope that you had a very nice long holiday weekend. If you were in the United States, we just celebrated July 4th. And to get us back on the ground for this week, we have our guest speaker, Dr. Brandon Henderson, who'll be talking about using electrophysiological methods to examine e-cigarette flavors' effects on dopamine neuron function. So I want to give a short background and intro for Dr. Henderson. So Dr. Henderson is an assistant professor in the Department of Biomedical Sciences at Marshall University School of Medicine. We call him Brandon, and he received his BS with distinction in chemistry at The Ohio State University, where he started research initially as an analytical chemist. He went on to receive his PhD in pharmacology at The Ohio State University in the lab of Dr. Dennis McKay. Then afterwards, his professional journey continued his development as an NIH postdoc fellow at Caltech in the lab of Dr. Henry Lester. And before starting his faculty position at Marshall University, Dr. Henderson also trained as a visiting postdoc fellow at Yale. And in addition to his responsibility now at Marshall, he is one of the two co-chairs for the Basic Science Network in the Society for Research on Nicotine and Tobacco. So do check out www.srnrt.org, and I'll put this in the chat box. So now the Henderson Lab focuses on the role tobacco and vaping flavors play in addiction-related behaviors. And this is a huge topic nowadays, as you may have seen in the news. And thus far, they have identified that two flavors, menthol and green apple, can enhance nicotine vapor self-administration and do so by directly altering dopamine neurons in the midbrain. So we're excited to welcome Dr. Brandon Henderson today to find out more about tobacco and vaping flavors and addiction-related behavior. So Brandon, I would love to hand it to you, and we look forward to your talk today.
All right, thank you for the opportunity, and thank you for the opening remarks. You make me sound smarter than I feel like I am, so thank you for that. So as Abby said, I'm going to talk about the electrophysiology that has been happening in the lab that supports some of our findings about how chemical flavorants in vaping products are altering behavior. And so to do that, so I'm going to start with the background just to orientate people. I'm not sure where people's background is or what their level of knowledge is regarding nicotine. So I'll start with the background, talk about the methods, and I'll talk sequentially about menthol and then green apple, specifically with what I've been finding with electrophysiological methods. So before I say anything, I'm gonna show some stats here, and I'll preface that this is with combustible cigarettes. So to date, combustible cigarettes are still the #1 cause of preventable death. That's around 500,000 deaths per year just in America. And, that may be supplanted by obesity-related disorders in the coming years, but still smoking tobacco products is the #1 cause of preventable death. This is mostly due to lung cancer. So lung cancer is one of the most aggressive and highest mortality cancer types, and tobacco products contribute to lung cancer, throat cancer, a number of cancers. And there's also a lot of illnesses caused by tobacco smoking and tobacco products. And this could be due to early exposure or in utero exposure that can cause birth defects. There's also incidents of asthma that are caused by first and second, and now they're finding third-hand smoke exposure can cause this. But some of the biggest problems are the fact that there is a number of people, about 75%, that smoke cigarettes. They want to quit, but they can't quit. And right now, the therapeutic options are relatively limited. There are a number of them with wide margins of success. And I'll mention that there's a lot of research into, so ENDS are electronic nicotine delivery systems, also known as e-cigarettes or vaping products. And some people are saying that this is a way of quitting tobacco smoke. And there's some studies that are showing that this may be a success. But the thing that we have to understand is, since vaping products have unique chemicals, there may be new health-related issues. So altogether, so part of our research is understanding some of these novel chemicals and what they may do as far as behavior and neurochemistry. So as I mentioned, there's this divide of combustible cigarettes and then vaping products. So popularity of vaping products, they're increasing over time, whereas combustible cigarettes have either plateaued or decreased over time. And this is actually, it depends upon where you live. So in rural areas, cigarette usage is staying stable, in some places increasing. So I grew up in Farmtown, Ohio, and still the smoking rates are high. It's part of the I guess you can say the environment that people are accustomed to. But in urban areas, this isn't the case. The rates have been declining over time. But the one thing that's unique about the vaping products is their usage of chemical flavorants to add flavor to the product. So if you look at the population of vapors, there's less than 5% of them that use a non-flavored product. And all of the places I've been to in the last 5 years, so California, I'm currently in West Virginia, I grew up in Ohio and I go there a lot. If I go to a smoke shop, I can't find a e-liquid that is not flavored in some way. It's really difficult to find. And there's flavors for almost anything you can think of. And I know you can't read some of these labels, but there's Waffle House flavors, or there's basically there's a Baconator flavor that I have a friend that really enjoys. And so any flavor you can think about, there's a product for that when it comes to the vaping market. And so I've been really interested in how these chemicals can alter behavior since they're inhaled, they have a unique route of delivery. Okay, so the other interesting thing is that when you compare combustible cigarettes to vaping devices, and I know that Juul is not the only pod-based system, but since it is the most popular and widely known, I always use that as an example. So the thing that makes this unique as far as nicotine and neurochemistry is that depending upon the device, there's different amounts of nicotine. I'll talk about why that's important in the next slide. So the tank-based systems, we have to all know that these are highly customizable. So the end user will fill these with their e-liquid of choice. And if your smoke shops are like the smoke shops in my neighborhood, you can customize that in the shop. So it's almost like a coffee house mentality. So if you want to mix a vanilla and green apple e-liquid, you can go to the shop, you can talk to the people running the shop, you can either buy them and mix it yourself, or they do it right there on the spot, and then you vape your newly customized e-liquid. So these are highly customizable, the nicotine concentrations are varied, and it really just depends upon what the user wants. So the pod-based systems, those are what we call closed loop systems. So basically you buy them, you can't modify them because it basically breaks the pod, and then you can't use it. There are some savvy people that can get around that, but typically you buy it and then you just insert it into your device and then you can vape that. So many of these have higher concentrations of nicotine, and they also use nicotine salts. And the importance of that is that the pharmacokinetic principles just allow it to have a faster a faster time to peak, bigger area under the curve. So it changes the profile of nicotine exposure. And the popularity of these devices have also made it so that the e-liquids that are used in tanks, they now have salt offerings of nicotine salt. So this has changed the market as well. So regardless of the nicotine type or the formulation, all drugs of abuse alter the dopamine reward system. Nicotine is no different. So nicotine will bind to nicotinic receptors in the ventral tegmental area. So there it'll alter these receptors so that dopamine transmission is enhanced, and then you can get the feel-good sensation. So I'll go through this really briefly because if you're interested in nicotinic receptors, there's tons of literature. And if you're not here to learn about nicotinic receptors, then this will just bore you. So I'll just mention that these are pentameric receptors, and their diversity is based upon the assembly of these pentamers. So there's two types. There are heteromeric types that are mixtures of alphas and betas, and there's homomeric types that are all alphas. But a number of these heteromeric receptors have been highlighted to be important for nicotine addiction due to a long history of research. So one of the first papers that talked about these receptors was published the year I was born, 1983. So this has been around longer than me. But receptors, nicotinic receptors were initially connected to the addiction of nicotine because these receptors are upregulated. And by that, it means that they increase in number. So this happens in humans, rodents, and cell lines. So this has been examined with human brain imaging. These are fluorescent reporter mice that were developed in my postdoctoral lab. But furthermore, so specific nicotinic receptors, we know they're important because if you train a mouse to self-administer nicotine, and then if you knock out either a beta-2, an alpha-6, or an alpha-4, those mice will then stop self-administration of nicotine. You can then selectively re-express these subunits in the VTA specifically. That will rescue the function. They'll start self-administering again. That doesn't happen in other brain regions. So this is just showing that these subunits are important and the VTA is important as well. So we don't use human models. So we use a mouse model. And our way of studying nicotine related addictions focuses on dopamine neurons in the VTA, but then also the projections that go in that are inhibitory. So there's GABAergic neurons, there's glutamatergic neurons that are excitatory, and then they send projections to nucleus accumbens. So we study that using several methods, but before I talk about that, I want to mention what nicotine does by itself before I can equate what the flavorants may do, because they can do things that are unique, but also similar. So typically, if we look at just the baseline of what's happening in the VTA, GABA neurons send inhibitory projections to dopamine neurons that will then go to the nucleus accumbens. So typically in a newly exposed to nicotine situation, both of these cell types will have nicotinic receptors that will then be activated, which will then have a net drive towards enhanced dopamine release, and you'll get some measure of a feel-good sensation. So nicotine isn't as rewarding as a stronger drug of abuse like cocaine or opioids. But so there's a mixture of reward, but then also just avoidance of the withdrawal related symptoms. But when a person is exposed long-term to nicotine, what happens is you have a higher degree of upregulation on these GABAergic neurons. This increases inhibition on the dopamine neurons, and this actually triggers craving because you have less dopamine being released over time. And then fast forward to the next time that person may smoke and have nicotine in their system. So nicotine will activate both receptors on the GABA dopamine neurons, but what will happen is these receptors on the GABA neurons, they turn off. We call this desensitization, and then you have a dramatic increase in dopamine tone, which then leads to a greater bolus of dopamine release, and this continues continued nicotine seeking. But then this is also complicated by excitatory projections that come from different areas of the brain. One of these I'll highlight is glutamate receptors that are in the medial VTA. Okay, so pretty soon I'll get to some data, but before I get to that, so why flavors and why is this so important? Because for decades, we just knew of, let's just say menthol as this is just a characterizing flavor. It just gives like a little bit of sweetness and it does nothing to behavior. But there's a lot of stats that kind of argued against that for a long period of time. So chiefly, I'll just focus on the fact that people that smoke menthol cigarettes compared to non-flavored cigarettes, they have a higher, they're less likely to quit. So their cessation rates are lower, and there's other associated stats. So like their time or the time to their first smoke, the number of cigarettes they'll consume. Basically there's a lot of stats suggesting that menthol cigarettes are more addictive than their non-flavored counterparts. And then Arthur Brody, who was a professor at UCLA, now he's in San Diego, he published a study in 2013, and this is what got me on this trajectory. So he looked at human brain imaging and showed that people that smoke menthol cigarettes have a higher degree of upregulation compared to those that smoke non-flavored cigarettes, which were then compared to people that were non-smokers. So this was showing that menthol enhanced upregulation, and this was one of the first neurochemical or neurobiological pieces of evidence that suggested that menthol isn't just a flavor, it's actually doing something to the brain to enhance addiction-related behavior. Okay, so that's the background and sort of what got me into this area. So now let's talk about the methods and how I conduct the research in the lab. Okay, So I'll start with what we're doing now, and then I'll backtrack to what we did to get us started. So right now, we're using a vapor self-administration system that is designed and basically assembled by La Jolla Alcohol Research Incorporated. So our system is amenable to mice. So the mice are freely moving. There's so they're in a closed airtight chamber with two nose pokes. One can be active, one can be inactive, and we can switch them depending upon the experiment. So we train them to nose poke for vaporized delivery of whatever e-liquid we're using. So we use the same tanks that are available at any type of smoke shops or anything that a person may use. We also have some adapters that we can use, pod-based systems, but essentially we're training the mice to vape just for the same e-liquids that a person may be using on the street. And we've gone through a number of different types of training paradigms to get the acquisition up so that it replicates what's happening in humans, and those are now published. So our first publication focused on using this method that produced about a 66% success rate with male mice, and it was slightly less effective for female mice. But we're currently using a method that is right now about 80% effective. We'll see as the coming months go, because this is a new method for us. But this has been published in our most recent paper. And so if you want the details of that, these are all open access and you can read about it. But what we've shown in our first publication, so this is a combined effort with my graduate student, Skylar Cooper. So we've shown that flavors enhance vapor self-administration of nicotine. So basically our tan bar or tan circles down here are nicotine by itself. And then our green and cyan colored are the combination of green apple or menthol with nicotine. So this is obvious that the mice are acquiring self-administration behavior at a faster rate and at a higher degree when the flavors are combined with nicotine. And one of the things that we found unique to this was that the green apple flavors by itself triggered acquisition of the self-administration behavior. So then once they're trained, we can then just see how much they're willing to respond for the different e-liquids. So here we also saw that green apple plus nicotine was enhanced versus nicotine by itself. And then menthol as well was enhanced versus nicotine. So basically saying that the flavors are enhancing acquisition and they're enhancing reinforcement related behavior in a mouse model. Okay. So right now our self-administration system, so we basically started this in late 2019, and we're just now getting to the point where we can use this model and then I can do electrophysiology. So the data I'm going to present from here on is actually using a previous model of drug exposure, which is conditioned place preference, and I'll show that right now. So this was started when I was still a postdoc and I was working with a graduate student who's now graduated and is now Dr. Tegan Wall. So she used conditioned place preference and then also trained me in this technique. But essentially what this is a non-contingent behavioral paradigm. So the mice aren't choosing to get the drugs, but we allow them to establish a preference in a 3-chamber system. And one chamber will give them a drug. So in this case, it would be saline, nicotine, nicotine plus menthol. And then we see as their preference changes, this can be indicative of reward if they basically move towards the drug paired chamber, or aversion if they move away from the drug paired chamber. So menthol by itself doesn't do anything. So we tried several doses and they basically had no increase. So an increase would be rewarding, a decrease would be aversive. So menthol by itself does nothing, but menthol plus nicotine enhances reward-related behavior. So you can take a non-rewarding dose of nicotine, you add menthol, it becomes rewarding. You take a rewarding dose of nicotine, add menthol, it becomes more rewarding. So from this data, we can say that menthol, it enhances reward-related behavior. It's not doing anything on itself at this level. It's only enhancing what nicotine is doing. So, from this point, the question is what's happening to the neurophysiology? And this is where we would take mice that were exposed to this paradigm extract their brains, and we would do the neurophysiology. And so this is where the electrophysiology technique comes into play. So just because I can't assume everyone is a neuroscientist here, I'm just going to explain that I mentioned that the receptors are important for nicotine's actions. So essentially, these control electrical signaling on the neurons. So chemicals bind to receptors. This triggers an electrical signal that propagates down an axon, which then repeats. And this is how neurons communicate with each other. And so we can study that using electrophysiology or electrochemistry. So in my lab, I do both. And so here's a picture of that. This is an old picture because this is one of my twins. He's now 3. He's much bigger, but he used to be attached to me when I would do some patching. This is actually the first cell I patched with him on my lap. So this is just showing that one of the keys of patching is just being able to see your neurons. If you can see your neurons, then you're able to patch. This is one of the reasons why, if you haven't listened to the previous webinar with Jonathan Ting, and you are an electrophysiologist, then you need to listen to that talk, because there's many people that haven't learned about his updated methods of brain slice preps, and it really improves things. So this just shows that we basically have a rig that allows us to microscopically analyze neurons. We then can custom fabricate patch pipettes that can then attach to a neuron, and we can study their electrical and chemical properties. So we use fluorescent reporter mice so that we can identify specific types. So here's an example of a mouse that was expressing tyrosine hydroxylase connected to GFP. So we can selectively identify dopamine neurons wherever they're expressed in the brain. This is an example of another GFP mouse. So we're looking in the medial habenula. So these are mice that are having fluorescent nicotinic receptors. And so we can select them specifically with the patch clamp electrophysiology. So as far as the method, so right now we've been using compresstome's VF300OZ. So we've used this. So since I started the lab, so this would be in 2017, we recently got to try out the 310, which was very nice, especially for people that are, since I've been doing this for a long time, setting the blade angle is extremely easy to me, and it takes about 10 seconds. But for a person that's never done it before, this model is great because you don't have to worry about the blade angle. It's pretty much all set. You can just get right to it. As far as slicing, so I mentioned the Jonathan Ting webinar. So he has a whole website that basically talks about his method. So people that have done patch clamp electrophysiology, I've always been trained to use what's called the protective cutting method. So people use either sucrose or glycerol-based ACSF as a way of protecting the neurons while they're cutting to minimize damage and minimize cell death. So I came from a lab, so all the labs at Caltech that I knew of were using glycerol-based ACSF, and so that's what I learned. But I've since learning about his method in about 2018, I've started using this protective recovery method. And just like he mentioned in his webinar, I've tried all the variations that are standard ACSF as far as the transcardial perfusion, or even not perfusing, just extracting the brain and then using the protective recovery method. And from my own experience, everything he said is totally true. The whole recovery step is critical. And then even there's been situations where I've completely botched the brain extraction and have only done the protective recovery method, and I still get a lot of good, healthy neurons. And I've been able to use this with mice that are 10 months old. And for those of you that have done a lot of patch clamp electrophysiology, you may be used to just, P30 mice or, something even earlier. But, this is a highly versatile method. I encourage you to look at this website, but then also the webinar that he did previously for Precisionary Instruments is really good. So I would encourage you to listen to that. So we're using basically a combination of his protective recovery method and what I use as the postdoc. So we're essentially doing protective cutting and protective recovery. So we use glycerol-based ACSF for the brain slicing, NMDG for the recovery, and then standard ACSF, and that's been working great for us. Okay, so here are some sample images from brains that we've sliced. So these are stained just to show that we use a alpha 6 GFP mouse. And the reason why we do that is because these have a 98.9% overlap with tyrosine hydroxylase. So I can kill two birds with one stone. I can select neurons that are positive for alpha-6 GFP and also know that they're TH positive and that they're indicative of dopamine neurons. And so I can do this for the VTA and I can do this for the SNC. Okay, so let's start with what's happening with menthol, and then eventually I'll get to what's happening with green apple flavors. Okay, so just as a preface, I mentioned before what nicotine is doing in the brain as far as the microcircuit of GABA neurons in the VTA that project to dopamine neurons in the VTA. So I mentioned that the nicotine will cause a disinhibition of the GABA neurons onto the dopamine neurons, and this drives dopamine tone. So looking at the same microcircuit, basically, you know, what is happening with nicotine plus menthol? And that's what I started addressing when I was finishing up my postdoc. So in this case, I was using a tyrosine hydroxylase GFP expressing mouse so that we can look directly at the dopamine neurons, and I can analyze basically THGFP positive dopamine neurons, THGFP negative, we presume are GABAergic neurons. There's additional tests we can do, and just in the interest of time, I did those and it's published. And so we could identify the dopamine and GABA neurons. So as I mentioned, nicotine will decrease the firing rate of VTA dopamine neurons. And part of this is because of the enhanced upregulation on these neurons, increasing inhibition. So when you add nicotine with menthol, what will happen is this effect is enhanced. And I can say it's enhanced because menthol by itself does something different. That's also in this publication and a previous publication. So that's complicated in a 40-minute talk on its own, so I won't mention it. Just trust me that I can know that this is an enhancement and not some sort of additive effect. So in the case of GABAergic neurons, nicotine is known to increase their firing frequency. Menthol added to nicotine further enhances that ability to enhance firing frequency. So what we see here is that menthol is enhancing what nicotine is doing and essentially making nicotine's job easier or better. So this is a baseline state. So what happens when you stimulate a nicotinic receptor? So essentially, I would patch a dopamine neuron, I would apply acetylcholine, that's the endogenous neurotransmitter for nicotinic receptors. And I'm using a high dose because this is basically going to trigger a peak response from the receptor. So this is kind of like an all or none type of effect. So I'll admit it's not physiologically relevant. We'll get to that in a second. But this is showing that if you stimulate the nicotinic receptors, we know that nicotine will have an enhanced firing frequency on dopamine neurons. But when you add nicotine plus menthol, what will happen is that this is further enhanced above what nicotine will do by itself. So this is just another example of menthol acting as an enhancer of nicotine's actions. So now the question is, in a smoking relevant situation, so a person that has been exposed to nicotine or nicotine plus menthol, you know, what will happen if you then apply a smoking relevant concentration of nicotine? So that was the next experiment. So another experiment, I know that this is the same neuron, but so we're looking at VTA dopamine neurons. So this is with a mouse that was saline treated, nicotine treated, or nicotine plus menthol treated. And then I'm applying a 10-second application of nicotine at a concentration of 500 nanomolar. And that's the peak concentration that would happen in the brain of a person that's smoking. When it comes to vaping, that just depends upon the type and the nicotine formulation. So that's a little bit more varied. But in a combustible cigarette smoking situation, this is the peak concentration of nicotine. And what we saw was that a nicotine treated mouse will have an enhanced firing frequency. This wasn't significant versus the control, but the mice that were treated with nicotine plus menthol had a enhancement versus what nicotine would cause. So once again, this is another example that menthol was enhancing something that nicotine was doing. So altogether, this is suggesting that there's several neurochemical reasons why a person may exhibit lower cessation rates when they're smoking a menthol cigarette. So in addition to what's happening with firing frequency, you know, we can ask the question about what's happening with channel activation. So the previous recordings are what we call current clamp recordings. We're looking at the firing of dopamine or GABAergic neurons, but we can also just look at what's happening with the receptor itself. And so for that, we use voltage clamp recordings. And essentially, we look at what's called a peak current amplitude. So this is indicative of how much ion flow is moving through the receptor. So it's been well established that nicotine increases the current density through nicotinic receptors, and this is indicative of upregulation. So both on dopamine neurons and on GABA neurons, we can see that nicotine will enhance peak current amplitude, but nicotine combined with menthol causes a further enhancement based upon the summary data. So in the dopamine neurons, it was not significant, but I'll tell you why in a bit. And but for the GABA neurons, it was significant. So what's complicated for dopamine neurons is that they exhibit a diversity of receptor types. So in GABAergic neurons in the midbrain, they only have alpha 4 beta 2 containing receptors, but the dopamine neurons have alpha 4 beta 2, alpha 6 beta 2, alpha 4 alpha 6 beta 2. It's complicated. So there's multiple different receptor subtypes, and this is why this is obfuscated, because you have to look at the receptor type to see what's happening. So one way of doing that is using a toxin called alpha conotoxin M2. This is selective for alpha 6 receptors. So essentially, the sensitive component is an alpha 6 receptor. So what we saw is that when we isolate that, there's no functional upregulation of those receptors. But if you remove that, essentially what you have is a alpha 4 non-alpha 6 containing subtype. So this means it's an alpha 4 beta 2. This is upregulated. So essentially this is now saying that menthol is subtype specific in its effect of changing neurobiology. So we can support this by using a cell culture model. So this isn't in a brain slice, but you can basically transfect specific nicotinic receptors into a cell model. And just to make it similar to the situation that we had in our mouse model that used fluorescent nicotinic receptors, we can do that same thing here. So we're using GFP labeled nicotinic receptors, and we're showing that the ones that are alpha 4 containing are enhanced by menthol and nicotine versus nicotine itself. And the ones that have alpha 6, they're not affected. So this is basically saying that menthol was supporting the fact that menthol is subtype specific in its effect. So here is a summary, and essentially it's basically what I've been saying at the end of all these slides is that nicotine will have an effect, but menthol is adds a further enhancement to what nicotine is doing, but it's also subtype specific. So I mentioned that it was having an effect on alpha 4 beta 2, not on alpha 6, but there's this more complicated subtype. And what's not shown here is that we can use a fluorescence microscopy to study the subtype that has an alpha 6 and an alpha 4. This is also upregulated as well. I just don't have time to talk about that because that's all microscopy. Okay, so that's what we found thus far with menthol. And so now we're focused on green apple flavors, mostly because next to menthol, they're one of the most popular types of flavors in vaping products. So this is this was as of 2019, a profile of widely used electronic cigarette e-liquids. So a large proportion, about 30% of the e-liquids used are fruity or floral. And there's a lot of intersection between what is fruity and what is floral. So there's many chemicals that are floral but are found in fruity containing e-liquids, and then vice versa. And so what happens is the green apple flavorants are among the popular next to menthol when it comes to vaping product usage. But the market's ever evolving, and this is something that keeps changing. So as of 2019, 2020, they were still extremely popular, and that's one of the reasons why we targeted them. So the popular chemical flavorants are farnesol, farnesene, methylbutyl acetate, hexyl acetate, and ethyl acetate. And I'll mention these last three are actually more popular than previous two. And we started with these two, and we're shifting our focus just with more up-to-date data of what's in products on the market. So similar to the menthol study, we started with conditional place preference because this was still at the advent of when we were getting our vapor self-administration model going. So my first tech, Austin, a medical student named Gabriella, who's now Dr. Casanelli, and my graduate student Skylar. So they all did studies using farnesol and farnesene in a conditioned place preference model. So what we saw with farnesol was that this was sex specific, and I'll mention this is a dosing effect. If you expand the dose range, we can see an effect with females. But at the initial doses we tested, farnesol caused reward-related behavior in a CPP model in the absence of nicotine. So this is unique from menthol because menthol didn't cause reward on its own. So farnesol caused reward on its own in a sex-specific manner. Farnesene did this or showed reward similarly to farnesol, but this was not sex-specific. So we saw this in males versus females. This is also a dose-dependent effect because you can see with the males, we're kind of at one side of what is typically an inverted U for behavioral responses, and we're probably just shifted slightly in one direction for the females where we can get multiple effects. It's important to know that if we use a nicotinic receptor antagonist, so here the example is DH beta E, we can block the reward. So this is due to a nicotinic receptor related mechanism. So now the question is what's happening with the neurophysiology. So similar to menthol, so we take mice that are exposed to the CPP paradigm. I'll take the brains out, I'll extract them, slice them, and do electrophysiology. So first up is with farnesol. So this is the green apple flavor that had a sex-specific effect. So here's some more up-to-date. So I'll mention the previous images I showed. Those are tail of my postdoc. I was using a different slicer. So I've always been happy with the current state of the health of the brain slices. And also, it's always nice to see great fluorescence and make sure you can get healthy neurons. So anyways, so what we saw is that identifying VTA dopamine neurons and looking at baseline firing frequency. So with male mice, we would compare the mice that went through saline CPP to the mice that went to farnesol CPP, and that in the male mice, they had an enhanced level of firing frequency, and this is at the baseline level, and we didn't see this in the females. So we chose the peak dose that we observed in males, and there was an effect that was absent in females, present in males. So similar to what we did with menthol, now the question is what happens when you add nicotine to the scenario? So here we're focusing just on the smoking relevant concentrations and smoking relevant physiology. So comparing a baseline state, so pre-nicotine exposure to the duration of nicotine exposure. So we're using a 500 nanomolar application of nicotine, and this is brief for 10 seconds, and looking at the state of the VTA dopamine neurons. So for the mice exposed to saline, we see an enhancement that's not significant. This was similar to what I saw with the menthol study. And my original hypothesis is that since the baseline state of the farnesol treated mice was already elevated, we would basically hit a ceiling. We wouldn't see that much enhancement, but that wasn't the case. These neurons fired much faster, despite the fact that they were already elevated. So this is one reason that can explain why farnesol could be rewarding, because it's just driving dopamine tone on its own. So the next question is what's happening with the GABAergic neurons, because remember, they're providing inhibitory input. So what I didn't show on the previous slide is that if you compare the saline treated to the farnesol treated at the baseline state, the GABAergic neurons are actually firing at a lower frequency. So that means a lower inhibitory tone. This partially explains why the dopamine neurons are firing faster, but then the nicotine exposure basically brings it to the same level of the non-exposed mice. So this is what this is all saying is that the net inhibitory tone is actually decreased in the mice that are exposed to farnesol. This partially explains why the dopamine neurons are firing at a much higher frequency. So that's basically what the summary is. The previous data I showed you was from the local GABAergic neurons in the VTA, but if you go to the posterior Some people call this the rostral medial tegmentum. So I've recorded from GABAergic neurons there, and it's essentially the same story. These have a decreased inhibitory tone. And this complements the drive towards increased firing frequency in the dopamine neurons, when, just as a reminder, will project to the nucleus accumbens. And this is one reason why farnesol is probably rewarding on its own. So the second green apple flavor is farnesene. And so using the same prep, so coronal slices, looking at VTA dopamine neurons, we identify them with GFP fluorescence. So I didn't see any change in the firing frequency. So that was unique compared to farnesol and menthol. But one of the things that was interesting about farnesene is that if you do a dose response of nicotine, what you see is that the mice exposed to farnesene, they have a leftward shift. So this is saying that the dopamine neurons themselves have a higher sensitivity to nicotine. So this is indicative of a change in the stoichiometry. So I don't want to get too deep into the biophysics of nicotinic receptors, but there's two stoichiometries of the alpha-4 beta-2 type, one that is a high sensitivity, one that is low sensitivity. And we postulated that there is more of the high sensitivity available after farnesene exposure. And we can do experiments to look just at that. So this is a FRET experiment. So you can ignore this part on the left. We'll focus just on this part on the right. So with the two different stoichiometries, you can do different FRET pairings. So the high sensitivity, if you use GFP M-cherry pairing, you can make it so that the high sensitivity will have 2 acceptors. The low sensitivity type will have 3 acceptors. And just with the nature of FRET, the more acceptors you have, the more energy. So when we saw a decrease in the energy after a farnesene treatment, this is supportive of the fact that we have more of the high sensitivity receptors that exhibit lower FRET energy. So this supported the finding that we're actually getting more of the high sensitivity receptors that are present after farnesene treatment. So this is suggesting that the neurons themselves are more sensitive to nicotine. To support that, we could look at what are called spontaneous excitatory postsynaptic currents. And so you can look at, so depending upon how you isolate the signal, if you were to block excitatory currents, you can then focus on spontaneous inhibitory currents, but we wanted to look at activation. So we blocked GABAergic signaling so that we can focus on what's happening with the excitation into dopamine neurons. And then what we saw was that the frequency of these excitatory currents, they're increased and the amplitude is increased as well. So essentially showing that farnesene treatment makes the dopamine neurons, I wouldn't say hyperexcitable, but the more excitable just due to the present excitatory stimuli that's in the VTA. So the other thing that's unique about farnesene is that, so I'll mention that in all the unique chemical flavorants I've always tested, I've done the full complement of control studies. You know, you apply them by themselves to see if they change firing frequency or if they stimulate inward currents. And to this point, I have never encountered anything that would stimulate a nicotinic receptor until we looked at farnesene. And farnesene by itself will actually stimulate inward currents that can be blocked by a nicotinic receptor antagonist. I'll preface that these are likely not smoking relevant because the lowest concentration I tried was around 5 micromolar. And so likely the smoking relevant concentration would be in the nanomolar range, maybe around 200 or 300 nanomolar. But farnesol or farnesene by itself will stimulate nicotinic receptor currents, which then classifies it as a partial agonist. Okay, so hopefully I've convinced you that, so with the earlier data I showed, so the flavors can enhance reward-related behavior. So this was in the CPP model. I didn't talk about that too much in detail since I wanted to focus on the electrophysiology, but our self-administration shows that the flavors enhance vapor self-administration. In the case of the green apple flavors, they can do this by themselves. I didn't show anything about upregulation of receptors, but we have several papers that show that green apple flavor can change the nature of upregulation. Menthol certainly enhances upregulation, But then this changes the landscape of the function of the receptors on dopamine and GABA neurons. And this is a principal component of why the electrophysiology is distinctly different. And one of the other things I haven't shown is the actual electrochemistry. So what does it do to dopamine release? So this has an effect on dopamine release. And just know that that's an important component, but I don't have time to talk about it. So at the end of the day, nicotine is known to modulate this dopamine pathway, but it seems that the flavorants can do this as well. In our case, menthol does this and the green apple flavors do this. And we're starting to look at other flavors involving vanilla, since that's also a very important vaping flavor. It shows up in a lot of e-liquids. So just to summarize, I want to thank my lab. So I'll mention that so far I've been the only person to do electrophysiology in my lab. So we're actually recruiting a postdoc specifically for EPhys, because I'll be free to admit that patching has been difficult since I became a dad. The great thing is that my lab focuses a lot on the microscopy and the behavior now, so that I have more time to do electrophysiology. So many thanks to them as well. And I'm also supported by NIDA. I've been supported by them essentially since I'm a graduate student, so I'm thankful for their support. And thanks for everyone for showing up. If you have questions about methods, I'd love to talk about that.
Well, Brandon, thank you so much for a great talk. And it was wonderful to meet the moral support of your lab too. At first I thought that these were the youngest electrophysiologists in your lab.
They actually have, well, one of them has actually patched a neuron. So I helped maybe like 80% of the way, but.
Oh, well, there you go. I was going to say they probably have more experience in patch clamp electrophysiology than the majority of graduate students in neuroscience, I have to say.
I'd like to say that, but I'm biased.
It's also wonderful to meet members of your lab too, especially Dr. Cooper's work on which you've just been presenting. I don't see any Q&A in the chat box, but for those of you who have joined us, please don't be shy about asking questions. At this point, also feel free to unmute yourself and also ask questions, and there's an open floor. So feel free to do that. Great. And we also will be posting a recording of our webinar online. So later on, if any of you who have attended have questions that pop up that you think of later on, you can contact us and we'll get your questions over to Dr. Henderson. And if there are no questions, we might end a little bit early, which I'm sure nobody will complain about. But it was a great talk by you, Brandon. Thank you so much.
Thanks for the opportunity.
All right. And with that, maybe then we'll close out and we'll post this. And again, thank you everybody for coming. And somebody is just saying thanks, Brandon, in the chat box.